Hello students, this is Dr. Anjani from Department of Biotechnology. Welcome to e-learning platform. Today we learn about structure of nucleus. Nucleus means kernel and was the first cell organelle to be discovered. It was discovered and named by Robert Brown in 1833 in the plant cells and is recognized as a constant feature of all animal and plant cells. It's an important cell organelle of all animal and plant cells. Certain eukaryotic cells such as the matured sieve tubes of higher plants and mammalian erythrocytes (RBCs) contain no nucleus. These are enucleated cells. It is the largest cellular organelle in eukaryotes. Prokaryotic cells lack nucleus or true nucleus and is complemented by nucleoid, is otherwise called as primitive nucleus. In mammalian cells, the average diameter of nucleus is approximately 6 micrometers, occupying about 10% of the total cell volume. The contents of nucleus are DNA, genome or genetic material, RNA synthetic apparatus, and a fibrous matrix. The nucleus is surrounded by two membranes. The membranes are composed of phospholipid bilayer containing many types of proteins. This is the structure of nucleus. In the cell, the nucleus is a membrane bound cell organelle. It's a membrane bound cell organelle having outer nuclear membrane and inner nuclear membrane, and it is not continuous. It is perforated here and there by pore like structures. These are called nuclear pore. This encloses a thread-like structure that is called chromatin reticulum, and it is associated with basic proteins, that is histone, and it is having a fluid-like material that is called nucleoplasm. And in the nucleoplasm, along with the chromatin reticulum, you'll find a densely stained structure that is called nucleolus. The inner nuclear membrane defines the nucleus itself. In most cells, the outer nuclear membrane is in continuous with the rough endoplasmic reticulum, and the space between the inner and outer nuclear membrane, it is called perinuclear space, is in continuous with the lumen of the rough endoplasmic reticulum. Rough endoplasmic reticulum will be surrounded by the ribosomes. The two nuclear membranes appear to fuse at nuclear pores. The ring-like complexes composed of specific membrane proteins through which material moves between the nucleus and the cytosol. So this nuclear pore provides the space for the movement of the material in and out of the nucleus. It contains the genetic material organized as multiple long linear DNA, and it is complexed with the histone proteins to form the chromosomes. The genes within these chromosomes are the cell's nuclear genome. The total number of genes present in the chromosome, total number of chromosomes represents the genome of an organism. The function is to maintain the integrity of the genes, and it also involves the controlling the activities of the cells by regulating the gene expression, either upregulating the specific gene or downregulating the specific gene at particular growth or at particular stage of cell development. In a growing or differentiated cells, the nucleus is metabolically active. The nucleus is active in the growing cells or in differentiating cells. What all it will do? Replicating DNA and synthesizing rRNA, tRNA and mRNA. Within the nucleus, mRNA binds to specific proteins, forming ribonucleoprotein particles. Within the nucleus, mRNA binds to specific protein, forming the ribonucleoprotein complexes or ribonucleoproteins. Most of the cell's ribosomal RNA is synthesized in the nucleolus, that is the darkly stained region inside the nucleus, that we call it as the nucleolus. That is the site for the synthesis of most of ribosomal RNA. This nucleolus is a subcompartment of the nucleus that is not bounded by phospholipid membrane. This nucleolus, it is important component of the nucleus. It is not surrounded by the membrane or phospholipid membrane. Some ribosomal proteins are added to ribosomal RNAs within the nucleolus as well. The finished or partly finished ribosomal subunits as well as tRNAs and mRNA containing particles pass through the nuclear pore into the cytosol for the use or for they have the role in the protein synthesis. So these tRNAs, mRNA and ribosomal RNA subunits will be synthesized in the nucleus. They may be completely formed or they may be partly formed inside the nucleus, and they pass through the nuclear pore at different time interval, and they reach the cytosol and they play an important role in protein synthesis. In a nucleus that is not dividing, the chromosomes are dispersed and not dense enough to be observed in the light microscope. So the cells which are not dividing show the chromosomes. They are dispersed throughout the nucleus and they are not dense enough to be observed under the microscope. Only during cell division, individual chromosomes visible by light microscopy. In the electron microscope, the non-nucleolar regions of the nucleus, called the nucleoplasm, can be seen to have dark and light staining areas. Under electron microscope, the fluid-like structure in the nucleus, that is the non-nucleolar regions of the nucleus called the nucleoplasm, can be seen to have in some region dark areas and in some other region you will find lightly stained areas. The dark areas, which are often closely associated with nuclear membrane contain condensed concentrated DNA called heterochromatic. Darkly stained region will be having condensed concentrated DNA called heterochromatic. The fibrous proteins called lamins form a two-dimensional network along the inner surface of the inner nuclear membrane, giving it shape and apparently binding DNA to it. Along the inner surface of the inner nuclear membrane, we will find the fibrous proteins called lamins. They play an important role in giving the shape to the nucleus and also provide the space for binding of DNA to the inner surface of the nuclear membrane. The breakdown of this network occurs early in the cell division. Before starting or prior to the cell division, there will be breaking down or demantling of this the lamins from the inner nuclear membrane. Inner nuclear membrane takes place. This is just an introduction to the nucleus. I hope you understood the topic. In the coming video, we will be learning about the ultrastructure of the nucleus. Thank you.